Buenos dias, I'm Juan Carlos Cambronero-Heinrichs, a PhD graduate from the University of Padova in Italy, and I will talk about host plant selection and performance of ambrosia beetles in flood stress trees versus ethanol injected trees, over two different publications from my PhD thesis. Ambrosia beetles are a group of weevils belonging to the subfamilies Scolytinae and Platypodinae. Although they are mostly native to tropical and subtropical regions of the world, they have recently emerged as highly successful invasive species. More than 50 species are now found outside their native ranges across the globe, with factors such as globalization and export of wood materials facilitating their spread. These insects are wood dwellers, spending most of their life cycle within the xylem of host trees, where they cultivate mutualistic nutrient-providing fungi carried in specialized organs from plant to plant. Many species are polyphagous, meaning that they can colonize multiple species of deciduous trees. Additionally, they exhibit highly developed social behaviors that support their fungal farming activities. Ambrosia beetles locate suitable host trees by sensing chemical cues. For example, opportunistic species of ambrosia beetles are known to attack seemingly healthy trees that emit ethanol. Abiotic stressors such as flooding and drought can trigger ethanol release. For instance, when a plant is exposed to flooding and the soil becomes saturated with water, oxygen availability is reduced, causing the plant to undergo fermentation. So, in a climate change scenario of increasing floodings, it's expected that ambrosia beetles will become more and more important. Other stressors, such as biotic factors, such as microbial infections, can also induce ethanol production and release. But overall, host selection by opportunistic species of ambrosia beetles is primarily driven by the presence of ethanol. Once a population of ambrosia beetles is established, their eradication is nearly impossible and has only been successful in controlled greenhouse environments. This makes it crucial to prevent their introduction into new habitats and to closely monitor existing population. In colonized environments, the only available option is population control. Various strategies exist, but one focus of my thesis was the use of ethanol injected trap trees, which are designed to attract and capture ambrosia beetles, thereby reducing their population density. In this, the first study I will comment, we use flooding as a stressor to trigger ambrosia beetle attacks. This approach allows us to investigate which plant species are more susceptible to beetle infestations and to identify which ambrosia beetles species can be expected to behave as more aggressive pests. We studied eight species of deciduous trees that are widely exploited in Europe. Four belonging to the order Fagales, including species used for wood production, ornamental purposes, and hazelnut cultivation. The other four belonged to the order Rosales, used for food production, including apples and pears. We located the plants in a natural forest where ambrosia beetles are known to occur, and to simulate flooding, we saturated the plants with water. After 28 days of exposure and beetle colonization, we cut the trees into logs, which were then taken to the laboratory and placed in reading chambers. We quantified the emerging beetles of different species, using this as our first response variable, a proxy for colonization success. After beetle emergence, we analyzed the logs using X-ray tomography, quantifying and identifying entry holes by species as a measure of host selection, our second response variable. Finally, each tunnel was classified according to its developmental stage, serving as a third response variable and an additional proxy for colonization success. In total, we observed attacks from four species of ambrosia beetles, two native to Europe, including Anisandrus dispar and Siloborinus saxeseni, and two exotic species, Silosandrus crassisculus and Silosandrus germanus. Across all response variable, it was evident that the exotic Silosandrus crassisculus was the most aggressive species. In terms of plant susceptibility, it was also clear that Rosales species were more affected by all ambrosia beetle species compared to Fagales trees. This aligns well with existing knowledge, as Silosandrus species are known to be a significant pest in apple orchards, for example. Additionally, within the same plant order, we observe species-specific preferences, suggesting that certain tree species may serve as more suitable hosts for ambrosia beetles. This first study was published in the Journal of Forest Ecology and Management. We concluded that the exotic Silosandrus crassiculatus is expected to be highly damaging species compared to the other species of ambrosia beetles in the study. Additionally, we found that Rosales species are more susceptible to ambrosia beetles attacks compared to Fagales trees, and that certain tree species may serve as more suitable hosts than others. In the second study, we investigated the effectiveness of using ethanol injected trees as traps to control ambrosia beetles populations. It is crucial to highlight that for ethanol injected trees to be effective in ambrosia beetle management, they must be more attractive than trees exposed to natural stress. Additionally, it is ideal that any deciduous trees used as trap has to be attractive, ensuring that ethanol injection can be applied to various tree species. This experiment was conducted alongside the previous one, using the same tree species, and after 28 days of beetle attacks, the trees were cut into logs and the same response variables were measured. When we analyze different response variables, for example, the number of entry holes, particularly for Silosandrus species, here in this slide, we observed a clear trend in fluted trees, where certain tree species were more attractive. For example, Malus sylvestris and Prunus armeniaca showed higher levels of attraction. However, when the trees were injected with ethanol, the numbers of attacks increased significantly, and the trends between different species become less distinct. In fact, ethanol injection flattens the host tree species preferences observed in natural stress. And this is ideal for management. This study was published in the Journal of Economic Entomology, and we concluded that ethanol injection can trigger more attacks than flooding stress, and that it nullifies the species-specific effects of different plant species. As a result, practitioners could use any species of deciduous trees as a trap for ambrosia beetles. With this, I conclude my presentation, and I will say many thanks to my supervisors, Professor Andrea Battisti and Davide Rasati, as well as my colleagues Giacomo Cavaletto and Giacomo Santorioma, who support made this project possible. I'm also grateful to Casa di Risparmio di Padova e Rovigo for awarding me the scholarship that supported my work. Thank you very much. Muchas gracias.